Broadcast is now starting. All attendees are in listen-only mode.
Well, good afternoon, everyone. My name is Jeff Smith, and I am Vice President of Public Policy here at AMIA. And I'd like to welcome everyone to another in the webinar series that we're started focused on COVID-19. Today, we're going to take a look at how another segment of AMIA's clinical research informatics community is contributing to better understanding the clinical nature of the coronavirus. Specifically, you'll be hearing from members of the Accrual to Clinical Trials, or ACT network, and they'll be presenting an overview of the existing network, which is funded by the NIH and NCATS, and will describe the goals of the ACT COVID-19 project. Presenters will discuss how they have been able to apply proven ACT technology, governance, and data use agreements to quickly implement a scalable COVID-19 research platform. And they will focus on COVID-19 extensions to the ACT query ontology, rapid implementation of a COVID-19 focused subnetwork, and their process to rapidly design, develop, implement, evaluate, and incrementally improve to meet the changing needs of the COVID-19 research community. Attendees will be able to understand how ACT fits into a variety of other efforts focused on COVID-19 research and how they can use the ACT and ACT COVID-19 project for their own research. Before we get started, I want to make sure that those of you who are unfamiliar, this is a webinar being presented by the American Medical Informatics Association. Health informatics is the science of how to use data, information, and knowledge to improve human health, including the execution of scientific research, the delivery of healthcare services, and the promotion of public health. AMIA is the multidisciplinary interprofessional home for 5,400 health informatics experts. Here is a view of AMIA's informatics community from the working group point of view. As I mentioned earlier, this is kind of a segment of the clinical research informatics community, but AMIA also represents the other domains, including consumer health informatics, public health informatics, translational bioinformatics, and of course, clinical informatics. So AMIA members really advance health and wellness by implementing and evaluating informatics interventions, innovations, and public policy across settings and patient populations, adding to our collective understanding of health in the 21st century through peer-reviewed journals and scientific meetings. So one way to think about informatics is through scales. We tend to think of patients as an N of 1 or 10 to the 0 power. Practices are usually measured in panels of 1,000, populations are in the millions, and globally we're talking about billions of patients. On the other end of the scale, we have thousands of diseases, millions of small molecules, and billions of lines of DNA. When we apply the scope of health informatics to this scale, it helps articulate the various overlapping boundaries of the 5 main areas of informatics. Translational bioinformatics professionals use informatics tools and methods that link biological entities such as genes, proteins, and small molecules to clinical entities such as diseases, symptoms, and drugs, or vice versa. Clinical research informatics professionals leverage IT and data systems to manage information related to clinical trials, and also involves informatics related to secondary research use of clinical data. Clinical informatics experts focus primarily on information systems that impact patient care and manage information related to their diseases and symptoms, and they do so at the practice level. Public health informatics is the application of informatics in areas of public health, including surveillance, reporting, and health promotion. Public health informatics and its corollary population informatics are concerned with groups rather than individuals. And then consumer informatics professionals analyze consumers' needs for information, studies and implements methods for making information accessible to consumers, and models and integrates consumers' preferences into health information systems. Finally, we can also think about policies on this scale. And perhaps the policy response to the coronavirus best exemplifies why the globe of health informatics is so important. Across all scales, from genomic to disease to patients and consumers, practices, populations, and the entire globe, informatics and AMIA members have a role to play. With this as a requisite background, and the informatics community is really the impetus behind the COVID-19 webinar series. So we have taken a look, I think this is our 10th webinar over the last several weeks, highlighting how various and specific groups of AMIA members and the informatics community are applying their specific domain expertise to the fight against the COVID-19 pandemic. For those of you who are unfamiliar, you'll be able to find more information on the COVID-19 webinar series at amia.org/COVID-19. And here you'll be able to download all the slides from the previous presentations, as well as hear recordings from the previous webinars. And like I said, we're getting close to double digits if we're not there already. And we have several more planned in the future. As the coronavirus continues to spread, and as we continue to learn more, there is a need to constantly update the world on really what the informatics community is doing in this fight. So with that, I would like to go ahead and kick things off over to the folks at the ACT Network to go ahead and take it away.
All right, well, thank you very much. And this is Lee Nadler speaking. I'm the PI of the Harvard Catalyst. Next slide. These will be the panelists. Okay, so we'd like to give a special thanks to AMIA for inviting us, and for the National Center for Advancing Translational Science for standing up and supporting ACT. So more than a decade ago, the I2B2 data warehouse and the Shrine search engine were born in Boston and shared open source. Individual CTSA sites implemented these tools to use the electronic health record to enable study of human health. Small networks across the nation were established, for example, the Harvard Shrine Network and the much larger UC Braid Network. Potential of the electronic health record to support human clinical research across the CTSA consortium was established. This network was launched in response to a national need. Individual academic healthcare centers across the CTSA network were struggling to meet the accrual goals of many of their important clinical trials. ACT, which originally stood for Accrual to Clinical Trials, we've changed the name now, was charged by NCATS to identify protocol-eligible patients using the electronic health record for cohort discovery. 7 years ago, ACT began with 4 sites, and through a series of managed waves, a total of 57 CTSAs are on or getting on the network. The next slide. So ACT's success is based upon team science. Although Steve Reese at Pittsburgh, Gary Firestein at UCSD, Bob Toto at the University of Texas Southwestern, and I serve as the PIs of ACT. The ACT leadership is truly horizontal and is operationally led by the ACT Executive Committee, composed of the PIs and all the work group leads. ACT is operationalized through its work groups and its network operations team. ACT responds to the needs of the CTSA hubs and their investigators, and is shown it goes bi-directionally, which speaks to the future strength and existing strength of ACT. Next slide, please. ACT work groups are shown in this slide, including governance, regulatory, technology, data harmonization, and data dissemination and evaluation. And I can say, having been part of this team, this is truly team science. Next slide. So the existing ACT network on this very complicated slide is depicted. It lists participating sites, and what these sites represent are approximately 130 million unique lives with 57 CTSAs participating. Next slide. The total queries, which initially were slow and as sites are picking up and increasing rapidly. And one of the things driving this is many of the sites continue to keep their small individual networks or small regional networks. And over the last year, those have been sunsetting and the work is picking up. Next slide, Doug will present.
Thank you, Lee. From sort of an overall design of how the ACT network works, our goal is to produce a network that has, as Lee mentioned, a large number of patients coming from a large number of sites. And we want that network to be as stable as possible so that all the end users, all the researchers at those sites can access it whenever they want. So here on the left side, we've produced that network. It's what we call the production network. It has a number of additional sort of features such as data stewards for monitoring behavior and getting people on the network. And of course, it is enabled for end user access, any researcher at these sites. We have two other networks. We have the stage network, which is where sites that are coming on to ACT will join there, make sure things are working right, and then they move to the production network. And lastly, we have the test network. This is a network where we test new software, new configurations, and so on before we move them to production. In this case, for the ACT COVID work, we reuse the test network, and I'll go into some details later about exactly what that new network looks like, the COVID-19 network. Next slide. Okay, so I think this slide has some animations. So yeah, keep clicking the animations and I'll sort of talk about it as it unfolds. So how the ACT network works. So each site, and like I said, there's over 40 of them in the production network right now. They inflate a local research data warehouse where they transition their EMR data into that research data warehouse. In this case, almost all of our sites are using I2B2. There's a few exceptions. That data is organized according to the ACT ontology, and Sham will be talking later about exactly what does the ontology look like and what have we done specifically to focus on COVID-19. In order for this research data warehouse to talk to a network, we put in the Shrine technology, which is basically a federated query network software, and associated with that are tools for people at that site to submit queries to that network. And so pretty much every site in our network looks basically like this, and users can submit queries. They go to all the sites and they get numbers back in a matter of seconds or a few minutes typically. Next slide.
Oh, it's back to me. So the uniqueness for all of you now that you're seeing just a big picture of the ACT network is what is truly different. The ACT network, the production network works that credentials users that can be any investigator at any of the ACT sites sit at their desk and run queries themselves. And over time, we've been noting that cohort identification was not driving what the needs of the investigators were. But considering that we have tens of thousands of investigators across numerous disciplines, the power of the network was obvious to us. So in the fall of 2019, ACT realized that it needed to support research based on the electronic health record. The challenge for us was governance, data use agreements, trust, and operationalization. In that late fall and early 2020, as we debated what kind of use cases we were going to use to set up the ACT research network, COVID-19 arrived. It was immediate that this was the perfect use case. And all of our groups, working groups, PIs, executive committee, committed itself to try to bring forth ACT to identify factors that would predispose individuals to infection, what factors would decrease risk. We'd attempt to identify factors that increase or decrease the severity of the COVID-19 illness. its outcome, and then we would try to identify repurposed drugs and sometimes drug toxicities. So as a group, we decided that we could not turn the entire ACT network into a COVID-19 network in one fell swoop. We decided to stand up the COVID-19 test network, which we are reporting to you today. So we convened our teams, and building upon our governance and data use agreements, we recruited 9 sites for the initial test network. 2 of them were at Harvard and University of Pittsburgh, UCSD, University of Texas Southwestern, UCLA, USC, University of Kansas, and UAB. And these sites all joined together. And I have never seen better team science, where the PIs and the informatics teams all melded. The working group structure was implemented. We disassembled the issues, made plans, set forth benchmarks. And over the last 2 months, we have brought all the sites on. We have, as you'll hear today, we've developed and revised the ontology. And as of today, I can proudly tell you that we've met not only our first goal, but our second goal. And today we enter beta testing. As you'll hear from my colleagues, we're ready to embark on a unique experiment to leverage the expertise and depth of the CTSA network to unravel many of the pressing problems of COVID-19 using real-world data. Back to you, Doug.
Thank you, Lee. When we identified COVID-19 as both an appropriate problem for us to apply the ACT network technology, as Lee mentioned, we have a very collaborative team model here that works with the working groups, and we have engagement at all of our CTSA sites, all the sites in our networks. So we rapidly formed a team of both sites that would be contributing to the COVID-19 network and members of the different working groups in order to determine exactly what we would need to do to make the COVID-19 network as effective as possible. We have a network inflated now with 9 sites. We are looking at additional sites to bring them on board while we test and validate this network. And we are augmenting the ontology. There's a number of new coded terms that relate to COVID-19, and we're also identifying other terms within our core ontology that might be relevant to some of the queries that people want to perform in the COVID-19 network. Of course, you know, data's only, querying is only as good as the data underneath. So we're rather extensively looking at the data that's in the network, doing consistency checks, data quality checks, and so on to identify any potential issues that may have occurred somewhere in the process. And then, as Lee pointed out, we're essentially today, we're at the point where we're recruiting small groups of researchers to get on and use the network and give us some feedback. We have a fairly iterative development cycle here for both ontology updates and adding sites to the network and some of the underlying technology components such as I2P2 and Shrine. So we expect this to be a highly iterative process as we go forward. Next slide. So the COVID-19 network that we've established has, as I mentioned earlier, 9 sites. I think Lee listed them out, but they'll also be listed later in this presentation. We have, I think, at least 5 more in queue to get on board. We have this concept of a data steward at each site that enables users from that site to get on board. Their function is somewhat like an honest broker, making sure that the users from their site are sort of following the rules of the network. We've drastically increased the data refresh cycles in this network. You may have noted on the previous slide about networks that in the production network, we ask sites to refresh their data monthly. given the rapid pace of the COVID-19 network, we realized that that was not going to be sufficient. So in this network, the goal is for each site that's participating to refresh their data at least twice a week. Some of them do it daily. As mentioned here, and of course later, we've developed new ontologies and done a few releases of that as we learn more about what's needed in there. We've revised our network agreement and some of the process involved with using the network and publishing from that. And then, of course, data quality in a variety of different ways, both to just maintenance of, is the network, the data in the network, of a high quality consistently going forward, and also as we put new elements in to comply with the COVID-19 ontology, making sure that those are well populated. Next slide. So the COVID-19 commitments from the sites, as mentioned in the prior slide, frequent data updates. So they've had almost all the sites had to significantly revise their ETL process from the electronic medical record. They have to be ready to accept new ontology and new ontology terms that changes their ETL process. Of course, they have to load this data on an efficient process. So we're not spending 2 days down to update the data and then go into another update cycle. And of course, as researchers get on board, everybody on the overall team needs to be available to answer questions. Next slide. And sort of lastly here, this is a quick snapshot of the network. We put together some information so different sites have different refresh cycles, and some of the sites were able to put in their whole population set into this network, and some of them, in order to meet those timeframes, put in a subset of that, still covering all the COVID-19 patients. As you can tell, across this whole network, the total number of patients involved are over 30 million. As of May 1, when we did some queries in the network, we have over 15,000 patients that have been tested positive for COVID-19, and overall 87,000 patients that have tested negative for COVID-19, for a total of over, it's like over 100,000 of patients who have had some test. Next slide. All right, I think this is Sham's bit.
Okay, thank you, Doug. So I'm going to talk about the ontology that we introduced for COVID-19, as well as a little bit about the cohort that we asked the sites to load. Next slide, please. So I'll touch upon the COVID-19 phenotype, the augmented ontology for COVID-19, some of the interesting features of the ontology, including the derived terms, as well as how we are prioritizing the derived terms for helping the sites map the data to those terms. Next slide, please. So one of the things that we realized when we started onboarding the sites was that some sites have a large number of patients in their I2B2 warehouse and won't be able to refresh this very frequently. So we provide 2 ways of frequent refreshers of the I2B2. One, if the I2B2 is small enough, then you can go ahead and refresh all data for all patients in the I2B2 on the accelerated schedule. Or if the I2B2 is large, then we provide a COVID-19 phenotype which identifies the patients which are of interest for COVID research, and only those patients are then updated on a frequent cycle. And we have a GitHub which provides this phenotype. Next slide, please. So this is a snapshot of the definition of the COVID-19 phenotype. It includes the ICD-10 codes which are relevant to COVID-19, both existing and new ones which have been introduced, CPTs, HCPCS, and LOIN codes. And as new codes are being assigned, we add them to the phenotype. In addition, we also keep this phenotype in sync with other initiatives like the CD2S initiative. Next slide, please. So this is a screenshot of the ontology that you see on the interface on the COVID-19 network. So it includes the standard ACT ontology, which allows you to query terms using demographics, diagnosis, lab tests, medications, procedures, and visits. And we have added this ACT COVID ontology on top of it. So you can use the existing terms from the standard ontology as well as the terms which are there in the COVID ontology. And I'll show you a little bit more about the COVID ontology. Next slide, please. So the COVID ontology is focused on kind of 3 types of codes. One set are the emerging codes. So newly introduced codes in ICD-10, CPT, HCPCS, LOINC, and which are therefore not there in the standard ACT ontology. We also include in it the existing codes from ICD-10 and CPT for convenience. And we have introduced a set of derived terms which are of particular interest for COVID-19 research. And this ontology is refined based on the emerging needs and is rapidly deployed. So we already deployed version 1, followed it up by version 2, and we are currently developing the next version of the ontology. Next slide, please. So this shows the next level in the COVID-19 ontology. So the terms are grouped into these 4 major categories. That includes diagnostic codes, course of illness, lab testing, as well as treatments. And on the right, this is the more exploded version of the ontology. And I just point out a few interesting features of this ontology. The first is a set of derived terms. And these look at the severity of illness of COVID, which is probably one of the most important things that you need to be able to query if you're doing COVID research. So for example, here we have intubation, mechanical ventilation, ECMO, supplemental oxygen, which requires some extra work in mapping those terms to the EHR data. In addition, we also introduced these harmonized terms, especially in the context of COVID labs, so that any COVID lab, the values are mapped to these 4 values, so that it makes it very easy to query, say, for example, all positive tested patients. And then a big part of the ontology is adding on the emerging codes, especially in the LOINC space. We also have added some of the existing codes which you can find in other parts of the ontology just for ease of use. These are especially in relation to medications that researchers might be interested in terms of COVID research. Next slide, please. So this just lists our prioritization scheme for the derived terms, because the derived terms take quite a lot of effort at the individual sites to map the data to these derived terms. So we're currently focused on these 11 derived terms, which are mainly in relationship to the severity of illness in COVID and the kind of service that might be provided, like inpatient service versus ICU. Next slide, please. Okay, so with that, I'll hand it over to Sean.
Thank you so much, Shyam. So, you know, if you look at the next slide, you'll realize that we really were trying to do something new in creating this highly dynamic system to ask new questions, where we'd have a COVID-19 centric portal allowing general institutional use with proper agreements in place with patient cohorts that can be studied, outcomes measured, and novel questions asked. And that was to enable the efficient and collaborative use of EHR data to answer specific scientific questions that arise throughout the pandemic using data of proven accuracy, possibly leading to the COVID-19 portal to perform feasibility assessments to guide clinical trials. So this is a new aspect of the ACT network, the old ACT network, now has a special significance in the study of COVID-19. And what makes it special for COVID-19? In the next slide, you see how many of the most relevant types of coding that's used to express probably the most fundamental fact about a COVID positive patient is very complicated. Here's all the different ways that a positive test in just one system for COVID-19 can be expressed. And that's because the pathology system actually has a free text entry that's used in order to express whether it's positive. Now, they try to focus on a few phrases, but at the end of the day, it's a pretty complicated scenario. Now, in most networks, that complicated scenario would be very difficult for the network to consolidate. If, for example, you tried to do it in a central location where you're devoid of local knowledge. But in the ACT network, because we have a very flexible system, which is highly dynamic, and we can utilize the extremely well-versed and super smart, I have to tell you, people at the local sites who know how to condense the data into, next slide, the single ontology item that we have in our new ontology that Sham described. So you see all that complexity is processed in the brains of our local informatics experts, and it is made into a fact in this highly dynamic I2B2 database, which really is a meta model and can have lots of different kinds of facts in it. So you make a new fact, essentially, which describes the fairly simple state of a patient having a positive COVID test. But there's a lot of work that goes behind that. And for many of you informatics experts, you'll realize this is trying to solve the curly brackets problem, right? So this is Ted's curly brackets problem that he described so many years ago. And we're just trying to direct, right, how we solve the curly brackets problem and allow the data to be put in there, which now can be used in further queries. And so if you look at the next slide, you see we also do the same thing to express COVID negative testing for this PCR test. And the next slide, you see we put together all of the different positive and negatives that we've derived from all the tests to make an even simpler code, right? Which is any positive COVID test and any negative COVID test. And if you look at the next slide, you see that once we've done that, that's a lot of work, but once that's done, we can ask questions that can simply go out as stored procedures operating on top of the I2B2 database, because we've effectively solved the curly brackets problem. And so now, if you look in the yellow box down towards the bottom, you see there is the primary keys for any positive and the primary keys for any negative test result. And so we can easily write a query, and this is the one that's right in front of you, in SQL. Of course, we know what the I2B2 database looks like, so we can write this query easily that answers the question, what patients have been COVID positive, more than 14 days went by, they were tested and became COVID negative, more than 14 days went by, and then they were COVID positive again. And many of you will think that, immediately recognize essentially, that this is a very important query, right? These are people who are getting COVID again, despite having been cured, so to speak, or their body cured them of COVID, they are getting COVID again. And this is a very important population of patients to look at. And it can easily be queried for once you have done this kind of groundwork in your ACT network. And this groundwork is all directed by the ontologies that are created in the ACT ontology team. Next slide. So once you kind of see how that can be done and think about it, you're like, wow, now we can actually do lots of interesting things. And that's exactly what happens. So now we have the same process where local smarts, right? These smart, bright people in the local I2B2 CTSA sites are able to think about, okay, so let's take one of them, mechanical ventilation. So how can we derive the fact that a patient's on mechanical ventilation? Well, actually, there's a lot of CPT codes, but a lot of times they're only coded after the patient's left the hospital. We want these updated every day. So that's not going to work. So there's some ICD-9 codes, but the same problem is going to exist. Another thing is there's flow charts, right? There's EPIC flow charts or Cerner flow charts, and you could derive it off of those and put it into your I2B2 database. Or you could be super clever and say, you know, there's some medications that are only used when people are on mechanical ventilation, like vecuronium, for example, right? It's a paralytic agent. And there's a whole slew of these. And so perhaps you can derive that those are somebody's on mechanical ventilation by looking at that. No matter how you reason it, it will be, you will condense it into this derived term. And this then can be used in further queries. And we do this throughout, including even whether a patient's dead, which can be pretty complicated sometimes in the way it's described and filters into databases. Now, the point here is that now you have these simple, I'll call them, facts about your patients, and you can answer all the pressing questions, interestingly. Next slide. So here's a couple of examples. So just from what you've seen and the rest of the ACT ontology, which exists in the background, of course, and people have spent months over the years making sure that, you know, they had medications, they had laboratories, they had diagnoses in there. You can say, okay, so we have a COVID positive patient who did particularly bad, right? They were intubated or they died. And what was it that predisposed that patient to that, perhaps? Was it some prior medications they're on? Was it some prior conditions they had? Interestingly enough, hypertension, the #1 prior condition. And what we want to answer now is, okay, is it that they have high blood pressure coming in, or that they just have the condition of hypertension, even though they might be on antihypertensives? So lots of interesting questions there. Another question, they're COVID positive, they got oxygen, right? What was from the laboratory, the PO2, the pressure in their artery of oxygen that led to intubation? If you have an art line, you're drawing oxygen levels all the time. So the question is, what is the level for therapeutic, for telling our ICU docs when it is that they should be intubating the patient? What is it that their oxygen level was that they had a particularly bad outcome? And the reason this question is very interesting is because it turns out that people with pneumonia from COVID-19 actually appear to do better if you delay their intubation. Their oxygen can go below 80%, which is against the mantra we have of emergency medicine. It can go below 80%, and people are still talking. In fact, they had to get some people off their cell phones in order to tell them they need to be intubated. This is counterintuitive, and we can show with this query that if you follow that practice, rather than letting them go down a little bit more, maybe into the 70s, they actually will do better and not die as much after their intubation. So that's a question. The third panel here shows, okay, so if you're COVID negative, right, and then you become COVID positive within 2 days, that means that there is something wrong with that COVID negative, that test, right? That test shouldn't have been COVID negative. You're not going to become COVID positive after less than 2 days. It just means that the COVID negative test was less, and therefore you can actually understand better the test sensitivity with that query. And then finally, there's something different about men and women. Women tend to not do as poorly with COVID disease as men. So how can we factor that out? We can factor that out not just by death, which has been done in a few papers, but also by oxygenation and mechanical ventilation need, which is what we have our derived facts for. So we can actually analyze this much better with the way that we've derived the facts. in the ACT database in terms of getting into the bottom of what is it about gender that is saving, so to speak, if you're a female. Is it hormones? Who knows? Okay, so next slide. So all this kind of just sets up the uniqueness of our ACT analytics, right? And I have to say, a lot of it is, it's unique because of all the hard work that's done by the bright, smart folks at the local CTSAs and that who help in building this network. It's a highly dynamic system to ask new questions because we can create new facts based upon the important features that patients have using the ingenuity of our folks at the local CTSAs. It's very respectful of patient and institutional privacy, distributing queries, not having the data, fly around. It distributes the queries, has them run locally, and then compiles the aggregate results. And I would say they're better, right, than compiling aggregate results as you have in some databases, because better than compiling just patient level data, because your local data expertise can be used to build facts guided by the ontology item definitions, solving the curly brackets problem, but at the same time being able to not lose the knowledge, right, that exists locally about what's important in that database and how it can be used, perhaps uniquely, to create the facts that we want are trying to direct in the ontology definitions. And then finally, much of this data, right, sets us up to be leveraged for clinical trials. So remdesivir, for example, what patients should we be giving that to? Is it patients who are getting progressively worse, or is it patients in the early course? And by having a lot of these derived facts, we can answer those questions and set us up for better clinical trials. Okay, next slide. I think that's also, oh no, this is still me. Next slide. So now let's just go into a little bit about how people have been leveraging networks overall in the US and internationally. Here's one, another I2B2 based one that was the academic users group, aggregated data from 96 hospitals, has a website, covidclinical.net, and has a preprint out where it showed over internationally how patients were having changing phenotypic characteristics as their disease progressed. Next slide. Here's a similar study done through OMOP, 4 hospitals, also has a preprint. And this one also looking at patients, and this one was clever in that they compared them with patients who had previously been hospitalized with influenza. Next slide. Here's a network that came out of New York. It's actually still in its formative phases. They actually had a New England Journal paper, but that was the local hospital that produced that work, not the network. And the interesting thing about this one is there's several different architectures. You can either send the data in, or you can do local aggregate queries and send the aggregation data in. And that's good because there's different times when you might want to be flexible with those 2 different strategies. And so that was a very interesting one. Next slide. Now, many of us are quite familiar with the N3C effort, which NCATS is hosting in order to amass a great deal of data in a single place that would then be used to answer COVID-19 questions. So a lot of patient level data being amassed. And then there's genomics and focused initiatives and sample focused initiatives like this host genomics initiative of you can see, and you can go to the website at COVID-19 HG. Next slide. Okay, so that's that concludes the comparisons there, and now we'll finish up.
Doug, you're going to take the last slide?
Yep, I'll take this. So go ahead and advance. Just to sort of cover in general how we expect this to be, the COVID-19 network to be accessed and so on. We're, as Lee pointed out earlier, we're in our first phase right now. We're beta testing. We're starting the beta testing process across a limited number of researchers in the 9 COVID-19 existing network sites. And of course, their work will be integrated with sort of our iterative process here. We'll learn from what works well for them, and what doesn't work well, we'll make some modifications. As we become more confident that that network is then really working the way that researchers would expect it to, we will increase the number of researchers from each of the COVID-19 sites. We're using existing processes for provisioning users and data stewards for keeping an eye on them. So this is essentially already built in. It's just a question of when we want to turn it on. And the same goes for the new sites that are joining. And then eventually we do want to push out much of what we've done in the COVID-19 network right into our production network, so the larger group of researchers can access it. There's a number of challenges with that, the frequent ontology updates and frequent data refresh cycles that many of our production sites may not be ready to do, but we are focused on doing whatever we can to get out into production. And so if you want to access the COVID-19 network or ACT in general, the primary requirement is that you be at a participating site. And so, if you're at a COVID-19 network site, then obviously eventually you'll be able to access that COVID-19 network. If you're part of an ACT network site, you'll be able to access that now. You've been able to access it for a long time. You may not know it, but you are. If your institution is not part of ACT, it's a good question. We were originally funded to support the CTSA consortium, so our focus has been on CTSA sites and their affiliated hospitals. And of course, if you're at those affiliated hospitals, you can also execute queries. If you're not a CTSA site, then right now the scenario would be to work with a collaborator that is at one of those sites, and then sort of by proxy, you can work with them and they can use the network and you can collaborate in that fashion. Over the long term, of course, we'd like to open this up and make the ACT network even bigger. That would be sort of a significant strategic step coming in the future. I think that pretty much is it for the prepared material. I think we should jump into Q&A.
All right, great. Well, thank you, everyone, for that really excellent information. For those of you in attendee land, you'll note that there is a questions box. I would ask that you go ahead and put your questions in there. We already have a couple, so I'm gonna, I'll go through and kind of group these as I'm able. We've got a couple technical questions. So first of all, let's see here. How many sites have successfully mapped to the top 11 derived ontology items, rather? And then another related question is on the new facts that are being created, about how long do those take? And can you talk about any of the, I guess, process to validate or verify those new facts?
This is Doug. I'll just jump in first. I think Sham can answer some of these questions in more detail. But this iteration of ontology, if I remember correctly, was initiated around 2 weeks ago. In that 2-week period, it's been the charge for the 9 participating sites to map to these new terms. And I believe they're substantially done now in order to enter the beta phase. Sham, feel free to chime in with other information.
Yeah, so I guess we have 9 sites on the COVID network at this point, and all of them have mapped to the derived facts in the current version of the ontology, is the version 2. And so we are now embarking on this beta testing to see how well that mapping has gone.
You guys should say, this is Lee, that This is an incredibly collaborative group. The informatics teams that we work with, there are 50 people on the calls. There are real questions. The harmonization group spends time alone so that it is team science. I want to stress that. No site is left to their own. And then, they compare best practices. And now when we beta test, we're going to find out what works and doesn't work. But the strength of our network is the depth and collaboration of the informatics community.
And this is Sean. I should just say many of you have been on some of our data quality calls, both the IDTF and the seminar that we had just a couple of weeks ago with CD2H. And what we do is we do a couple of smoke tests. One is we just use the terms in order to figure out if things look right across the network, and we can do that because we have our network query tools and we can test different combinations of logic. Second, we actually query for the numbers of the query term of these derived terms, and just to make sure they look right. We have an idea of how many terms should be at on average at each site, given the kind of hospital that it is, and then we'll look for that. And then third, at a few selected sites, we go deep into the record to look to find out if the terms are working appropriately at sites by actually looking in the record. So kind of a three-phase validation process.
Okay, great. I've got a couple more coming in here. So a couple of questions about, I guess, kind of getting existing sites plugged into the COVID network, and then potential new sites. Is there specific contact information that folks should reach out to if they're, I guess, a part of a local network versus not part of the ACT network at all?
So this is Lee. I'll take a shot at that. I've been the person putting the sites together, and we are very much interested in bringing sites in. We've teed up University of Seattle and Washington, UCSF, University of Michigan, University of Pennsylvania, several others. If anybody wants to join the network that's part of the ACT network, please reach out to me and we'll go over, before I have to hand you off to the informatics team, I'll go over what's required. And I'm willing myself to speak to your PIs and to the people at your sites, because that's been my approach, to build the team, not just from informatics, but from across the leaderships of the CTSAs. If you're not part of the network, it's more difficult and you'd have to find a collaborator.
Okay, great. And we'll make sure that your contact information, Lee, is included as part of these slides. And then again, for those of you who are wanting access to the slides, they'll be available on the AMIA website tomorrow. And then we will distribute sometime next week the full recording and slides in case you haven't had a chance to make it back to the website. I've got a couple additional questions here focused on, let's see here, focused on the kind of plans for publication and dissemination. It seems like there's a lot of really interesting, and as you said, dynamic research questions that can be asked of these data. And given the fast-changing nature and I would say kind of the stress that traditional scholastic and academic publishing has gone under in these last several weeks, do you have a sense of when you think some of these questions you're asking are going to be ready for publication and or dissemination?
I'll take this one again. The PIs today form the nucleus of the publication committee, but we're reaching out to get representatives from all 9 sites. Publication committee should not be top-down, it should be horizontal. Depending on, we do not want to be a filter on what the type of question is. We do want to make sure that it is respectful of each other's CTSAs. that it follows our governance documents and data use agreements. And we're going to work very hard to bring people together to work together as teams. So we have a list of about 15 really important areas for COVID research. I'm sure there's 20 more, but we made a list of about 15. And we, in our previous call earlier today, we're really eager to put teams together of people to work together. So instead of having, I mean, anybody at any site can do what they want, but we think if we bring the smartest people together, that will show the strength of the CTSA network. And as we've watched everything else in the nation, where every state is trying to do its own thing, we'd rather not. We'd rather be not red, blue, but the United States working together to help our patients. So, and anybody who has thoughts, write to us. We would love to hear how you think this should be done. We're all ears.
Okay, great. I think we've got time for just one or two more questions here. This is another, I think, another kind of process or timeline question, and it really is asking around how long does it take to bring the kind of ontology into production?
Sean, Doug?
Okay, I can talk about this. So the ontology releases, so the ontology is put together mainly at the University of Pittsburgh. We take in all kinds of input, put it together and release it. And so the update at any local site is typically very fast. You can do this within minutes. And most of the time, the update just involves updating just the COVID trunk. So you don't have to update the rest of the ontology.
There is, of course, if there's new terms added, there is, of course, for those to be properly presented for query, the local site, each local site needs to make sure they map properly to that, as Sean and Sham have mentioned regarding both derived terms and new COVID terms.
Okay,
Yeah,
great.
that's been taking a couple of weeks on average. It's been taking about a couple of weeks to do the new terms, which was pretty amazing, actually, considering, you know, how complicated some of this is. But people have really stepped up. Really stepped up. It's amazing. Really amazing.
Excellent. Well, thank you so much for all of that great information. And for those of you who are still with us, we've still got a great number of attendees from the beginning. I think you all have, like I have been, kind of hooked to the screen and listening to a lot of really excellent work being done. And I think this is, as the presenters noted, you know, this is a good time to be in informatics and a good time to try and flex some of those muscles in pursuit of the national imperative to try and address COVID. I will just close off by saying thank you to everyone who attended and to remind those of you who are interested in the clinical informatics realm that we are having a virtual clinical informatics this year. You'll find information on registration and agenda at that website. And rest assured that this will not be the last COVID-19 webinar. We will have a couple more that we will be promoting over the next few days that will be coming to you next week. I believe a global health informatics look on what low and middle income countries are doing to establish informatics infrastructure and fight COVID-19, as well as additional webinars focused on this, which is the clinical research informatics angle from the NIH and NCATS. So thank you so much, everyone. Have a good rest of the work week and talk to you soon.
Bye all.